Welcome to the lesson number 9 of our module. In this in this chapter, we are going to learn about the validation and what validation is, how it is important, and how pharmaceutical analysis is used in the validation techniques. So, not making more delay, let us start our lesson number 9. Lesson number 9, Method Validation. Validation and quality control in pharmaceutical analysis. So dear students, in this lesson number 9, we will focus on the critical aspects of validation and quality control. We will understand how validation is associated with quality control, and we will also understand what method validation is. So the main thing that we will revolve around in this chapter will be the method validation. See, as you know that we are using different analytical procedures to identify or to quantify different pharmaceutical substances. Now, these methods have to be extremely reliable, and these methods also needs to be extremely accurate. And to ensure that these methods are reliable and accurate, we perform validation analysis or we perform method validation. There is not much to say that these methods or the method validation is very important for the pharmaceutical research, development, and the manufacturing process. Now let us understand the importance of method validation. As you see that we are performing analysis in day-to-day pharmaceutical processes. So as we are performing different analytical techniques and analytical methods, we have to ensure that these methods are accurate, these methods are precise, and these methods are extremely reliable. Accuracy means that how much our experimental value is close to the true value, the actual value. And precision means how much our experimental values are close to each other. Reliability, obviously if accuracy and precisions are there, our method reliability increases. We have to check whether our methods are reliable in different conditions. Are they giving same results? Are they reproducible? Are these procedures robust in nature or not? So we have to ensure that our analytical methods are reliable. So obviously if methods are reliable, the results which are the outcome of these methods will also be reliable. Second point of importance is it validates, it ensures us, it tells us what method validation. Method validation validates or ensures or tells us that particular analytical methods which we are using is suitable for the intended purpose. Let me tell you one thing. There can be more than one method. Method 1, method 2, method 3. There can be more than one method for a particular kind of analysis. But the best method is selected by the method validation process. Notably, this is a very crucial step because this method validation is an important part of the regulatory requirement. And if the correct method is used and if method validation is done properly, that ensures the quality, and obviously that is why method validation is an important part of the regulatory requirement and the industry standards. Method validation also ensures the quality of the product, but indirectly. Now what are the parameters in method validation? There are following parameters. Number one is accuracy. I have said that accuracy is the closeness of our experimental results to the true value. How much an experimental value is close to the true value. The more it is close, the lesser is the difference between true value and experimental value, the more accuracy our method is having. So we have to check for the accuracy, and it is good to have higher accuracy in our method. Second parameter is precision. Precision means how much two experimental values are close to each other. See, we have done an experiment analytical method. Now we have got experimental value number 1, experimental value number 2, and experimental value number 3. Now if these are very close to each other, so that means our method has reproducibility. Reproduce is very important. If method does not have reproducibility, then it is not counted as a good method. Third thing is specificity. Specificity means that our method has the ability to detect the analyte of our interest and not the other component present in the matrix. So it has to be specific. Like for example, in a sample, if there is an ion A, an ion B, and another ion C, I just want to detect only A. Then our method should be such like that it only selects A. That is why it is having the specificity. It detects only A, that means our method is specific. Now linearity. In the previous chapter, in the previous videos, we have already discussed about the standard curves, calibration curves. The linearity is related to this. See, there is a concentration of the pharmaceutical substance or the sample of the substance in the sample, and there is a response given by the instrument. So we expect that when the concentration is increased, let us understand that, for example, the x-axis here, it tells us concentration, and y-axis tells the response of an instrument. Now we expect that as the concentration increases, the response increases linearly. It has to be linear. Why it has to be linear? Because if it is linear, then it will be easier for us to identify the unknown substance. Suppose that this is the graph, and let us say that we get a response at this point of an unknown sample. Now what we will do is that we will draw a parallel line to the x-axis. It will cut this graph, linear graph, at this point. Now we will produce a perpendicular onto the x-axis. Now we can easily tell us the corresponding concentration, and this concentration will be the concentration of unknown sample who has given this response y. So linearity is extremely required, because we can do this thing only in case when there is linearity. In the later classes, you will understand that linearity is calculated by the R square value. So these are 4 parameters which are extremely important in the method validation. These parameters will say that what are the points that we are considering while performing method validation. What are the features of our method, our analytical procedure that we are wishing to see. And these things are accuracy, precision, specificity, and linearity. Now, as there are different guidelines in pharmaceutical industries according which we should be doing our procedures, that will ensure the quality, safety, and efficacy of our substance. These all methods, they have to be performed under some specific guidelines which are called as GLP and GMP. GLP basically works for the laboratory practices, and GMP are the guidelines for the manufacturing processes. So these are the good laboratory practices. One has to comply with these practices in order to get the approval of regulating body. The GLP are the guidelines basically for conducting non-clinical laboratory studies. Obviously, it will ensure the data integrity, reliability and traceability. Because if you are performing good laboratory practices, your data integrity will be good, the reliability of your method will be good, and you can also trace back if any problem arises. Now, good manufacturing practices, GMP, these are the standards for production and testing of pharmaceuticals. Obviously, they both are the part of manufacturing processes. If GMP are followed, obviously it will emphasize the quality control document. It actually ensures quality control. So GMP emphasize on quality control. It emphasize that proper documentation should be done, and it emphasize on the consistency of our methods. Now, about quality control and quality assurance in pharmaceutical analysis. Now, dear students, these two terms are very important, and most of the time student gets confused during between these two terms, but you must understand what these are. The first is quality control. So, quality controls involve monitoring and ensuring the consistency of the analytical processes. See, in a simple word, you should understand that quality control is nothing but in process testing. You are testing during the process, and why you are testing? You are testing to ensure the quality, ensure that your quality is good. So it's a proper action thing that you are doing. You are constantly checking during the process of manufacturing. So how will you do that? You will check the quality of the sample by using standards and the reference material. But when you are doing quality assurance, you are actually assuring that whatever process you are doing, this process will lead to meet the defined standards. Like for example, when you are doing manufacturing, you will start from the raw material. You will take the raw material, then you will process it, you will do the manufacture, you will do the packaging and other things. So in quality assurance, there will be some guidelines, there will be some procedures that you need to follow. They are already set procedures that you are performing. Like for example, if you are bringing the raw material should be of this quality. That standard is given by quality assurance. Your process should be follow this particular thing. This is done by quality assurance. Your packaging should be of this type. This is done by quality assurance. And quality assurance constantly checks, audits these processes, the raw material quality and all the steps in the manufacturing. They are also validating that whatever happening, whatever the standards are given, they are being followed properly. Now, quality control and quality assurance, if I compare these, so we take quality assurance as a broader term, and quality control is actually a subset of quality assurance. Quality control is just that part of quality assurance which is performed during testing. But quality assurance is performed before the manufacturing process and even after the manufacturing process. Quality controls is just telling that everything which is quality control will just test them while the process is going on, while the manufacturing process is going on. So these two terms are very important. Let us conclude them again. What is the big difference between quality control and quality assurance? Quality control is basically the procedure to check the quality in process, whether whether the product is formed in a good quality or not. And quality assurance is covering all the steps starting from the raw material up to the end, that the product of the desired quality is being formed. And how they will check it? They have already created some standards, some procedures, and you have to, are the scientist or the person in the manufacturing process who is involved, are they are following these processes or not? So they are constantly checking. A typical example in our daily life, we can also understand quality control and quality assurance. See, when say you are cooking meal, you are cooking your meal, and obviously when you will cook your meal, during the cooking you are tasting it, you are checking the consistency of your food, you are checking the color of your food, you are checking that it should not be overcooked or it should not be undercooked. So during process, whatever you are doing to control the quality, to make it a good food, these all comes under quality control. But what is quality assurance? Quality assurance start from the beginning, where you have brought your raw materials for cooking. You will ensure that all the vegetables or all the food material that you are bringing should not be stale, should be of good quality. All the spices that you are making, they are you are bringing, that should be of good quality, because you are assuring, if your raw material is of good quality, obviously your product, your food, your meal will become of good quality. And then you will also ensure that your utensils are cleaned in which you are washing. You will also understand that after preparing the food, it should be served properly. So everything under it, including the tasting, including the cooking process, including the quality control, they all come under quality assurance. So this is a very simple example to understand quality control and quality assurance difference. Now, analytical instrument qualification. See, it's very easy to understand. During analysis, most of the time we are doing it with the help of an instrument. So obviously you need to assure that your instrument is of the good quality, it is capable to perform the analytical procedure. So when you are testing this instrument to check whether they are fit for doing the analytical procedure or not, you are doing quality instrument qualification. So analytical instrument qualification, in short known as AIQ, it ensures that analytical instrument that we are using for the pharmaceutical analysis, they meet predefined specifications. There has to be some standards which are laid down and that our instrument should follow in order to get the accurate analytical results. These instrument qualification have some part, and you must know these terms: installation qualification, operational qualification, and performance qualification. These are very important terms that are frequently used during instrument qualification. So installation qualification is at the time when you are installing the instrument. Operational qualification you are doing while instrument is performing its work. And last, you are doing the performance qualification. You are constantly checking your instrument is giving the quality results or not. Now the regulatory compliance are there. You are constantly complying with the regulatory bodies. You have to adhere with the regulatory guidelines like FDA, EMA, and these are very crucial for the pharmaceutical companies, because if you are not complying with the regulatory guidelines, you will not get the approval to sell your drugs. Now, obviously, non-compliance will lead to regulatory actions. Product will be recalled. Recalled means you have you have distributed into the market and then you are bringing back from the market. There will be a fine imposed to the pharmaceutical company if they are not complying with the regulatory standards. So what are the key elements of quality control plan? So the key elements, the most important points of quality control plan are SOPs. SOPs are known as standard operating procedure. To do any work in your laboratory, there are SOPs written. These are the specific methods that you need to follow while doing something. There are SOPs for the procedures, there are SOPs for the instrument working. So these are the detailed instruction for all the laboratory processes. Now calibration and maintenance. To perform the quality control, you have to regularly calibrate and maintain your instrument. Instrument calibration is the most important thing, because instrument calibration ensures that our instrument is measuring the desired values. Now, training and competency. Obviously, everything, whatever we are doing, either running an instrument or doing any process, that has to be done by the personnel, and that personnel should be trained. If the person is not trained, it will arise to systematic error. We don't want these kind of errors to happen, because they will affect the quality of the product. So to control the quality, we have laid down SOPs, we have instrument calibration and maintenance, and we are also training the personals. Now let us summarize our chapter. So method validation is very essential. Why? Because that ensures the accuracy and reliability of analytical methods. The guidelines like GLP and GMP, they establish the standards for laboratory and manufacturing practices. Quality control and assurance are very important, because they maintain consistent and high quality of the product. I have said that quality control is basically dealing in process, and quality assurance is covering everything including quality control. Now you need to comply with the regulatory standards. If you don't comply, you will have to face the consequences, and that's all about this chapter. Thank you. So hello everyone, I hope you have understood the lesson number 9, which is the validation techniques. And in this chapter you have studied about the good laboratory practices and good manufacturing practices. So as an undergraduate student, you should understand the basic concepts of the same. Our next video will be on the real time application of pharmaceutical analysis. That means you will study how pharmaceutical analysis is being used, and you will study some of the case. So till then, thank you so much, and we'll meet on the next video.